This is a cross-section of an onion root. So these are onion root tip cells, and you can see that many of them are going through mitosis. You can see the chromosomes in some of them. So why all this mitosis going on in onion root tips? Well, that's where the root is growing down into the soil. So what you can see right here, these are individual cells, and here is the nucleus and the nucleolus on the inside. And some of them, you can see that the nuclear membrane has disappeared, and you can see the chromosomes inside. And there are different stages of mitosis going on here. So let's take a look at mitosis in a little bit more detail in this drawing right here. Let me get my pen. Okay, so here is our cell, and it's in G1. So we have 4 chromosomes. So 2n equals 4. So it's 2 sets of 2. So here are the chromosomes from mom, here are the chromosomes from dad. And right, we call this diploid. And organisms have different chromosomes. All species have different numbers of chromosomes. We have 46. Some might have 26. Some might have 20. Some might have 4. So this organism right here, the somatic cells, have 4 chromosomes. So the idea of mitosis is to have cell division with the same number of chromosomes as the parent cell had. So in S phase, we know that DNA is replicated. So let's identify here. We have, this is a chromosome, this purple chromosome from mom. This, and oh, what color do you think that is? Orange. Let's say orange chromosome from dad. So they look similar. They're the same length. They're not exactly the same because they have some different nucleotides in the DNA strand, but they are similar. So these are called homologous pairs. And these two are homologous pairs. So let's put that up there. So homologous pairs. When the DNA is replicated in S phase, so here is one chromosome. So this chromosome right here was replicated in S phase. That means that there is another exact copy of that chromosome, and they are attached, those exact strands of, the same strands are connected in what is called a centromere. Centromere. And then each one of these is now called a sister chromatid. So also in S phase, these things right here, they are called centrosomes. Goodness, a lot of centros. So these are centrosomes, and they are duplicated. So centrosomes. Okay, we good with S phase. Now we're actually going into mitosis from this point right here. We're going into prophase. So there are some events that are happening here that identify this as prophase. One is that the nuclear membrane is starting to break down, and so that you can see the chromosomes. The chromosomes are changing from chromatin form to chromosome form, which means they're getting all folded up and bundled up so that it's easier to move around the cell. And this drawing is trying to show that. I mean, much thinner over here and then much thicker over here. So these are chromosomes. This is one chromosome right here. It has its sister chromatids, and then both of these together are a homologous pair, still a homologous pair. Okay, and another thing that's happening in prophase is that these centrosomes are moving to opposite poles of the cell. Okay, now we move on to metaphase. And defining characteristic of metaphase is that the chromosomes line up on what's called the metaphase plate. And these things come out right here from the centrosomes are called spindle fibers. And they attach to the chromosomes at the centromeres at what's called a kinetochore. So there is at the centrosome a kinetochore on each sister chromatid. So kinetochore. And these spindle fibers will then shorten. And as they shorten, it pulls the sister chromatids apart. So the sister chromatids are going to be pulled apart. And here they are in anaphase. So in anaphase, these sister chromatids are being pulled apart. And here's the centromeres. So all chromosomes have a centromere. Even in the parent cell, there are centromeres here. So that has a centromere. You know this is one chromosome because it has one centromere. So these are now daughter chromosomes because they've been pulled apart. So they are now daughter chromosomes. These spindle fibers are actually coming apart at the centrosome, and that's why they shorten. But there are a few, and I lost my pen. How did I do that? There are a few that actually lengthen instead of shorten. And what they're doing is they're coming together and they're pushing the cell apart and lengthening the cell. So they're pushing it apart and lengthening it. So that in telophase, now you've got your chromosomes at different poles. And telophase, you tend to see the chromosomes all bunched up at each pole, and the nuclear membrane starts to form again around them. And at the same time, so that's mitosis, at the same time, the rest of the cell is dividing as well. All those organelles that had been replicated are now going into each part, or each individual cell now, to each pole. And in this case right here, what's happening is that the cell membrane is being pinched in. So this is an animal cell, whereas a plant cell, the cell wall is formed between each new daughter cell. In an animal cell, the cell membrane just sort of gets pinched in. Okay, and so through mitosis and through cytokinesis, we now have two new cells with exactly the same number of chromosomes that we started with. Okay, so this is a cycle. So these cells can now go through mitosis again, cell division and mitosis, or they can go off into what's called a G0 phase. And that's pretty much their job. They can go off to do the job that they're intended to do, say such as our skin cells. So here are the cells that are going to divide. When a cell gets pushed along out to the outer surface of the skin, these cells out here are no longer going to divide. They're not going to go through mitosis. They're going into G0 to do their job, to be skin, but they are no longer going to go through cell division. Okay, so that is mitosis, and I hope you found that interesting, and let's move on to meiosis.